hardest part of gene therapy isn't making genetic edits. It's getting enough of the editor into the right cells. If you infuse a patient with gene editor particles that target, say, muscle cells, you'll be lucky if 20% of muscle cells get a particle. This is partly limited by dosage, but if you push the dose too high, you start getting toxicity. So this paper tests out a clever new strategy. Instead of trying to deliver more CRISPR, what if you make the small number of recipient cells propagate the signal? The approach, called NANITE, turns initial recipient cells into temporary factories that produce and send out gene editing enzymes. Here's how it works. Initial recipient cells get DNA instructions to assemble CRISPR complexes, package them into little capsules, and send them out into the surrounding tissue. Those capsules can then fuse with neighboring cells and deliver the editor directly, even though those cells never received the original therapy. Conveniently, these CRISPR-containing particles can be targeted to specific cell types as well. The authors nicely demonstrate NANITE in mice, targeting a disease-related gene in the liver with CRISPR knockout. They see double the amount of editing with NANITE compared to delivering just the editor, and that significantly improved the therapeutic outcome. So really promising strategy overall, but limitations? Well, first of all, it's hard to efficiently deliver these big bulky DNA instructions, even with a lipid nanoparticle, which is unfortunate when the whole point is to boost delivery efficiency, but maybe using mRNA instead of DNA will help with that. Second, if used therapeutically, would there be any safety concerns with the expression of all these particle proteins? The transience of mRNA versus DNA might also help with this, but obviously it's still something to test. Still, the core idea here is powerful. Rather than asking how to deliver gene editor into every cell, NANITE asks how much work can each successful delivery do? And pairing this sort of amplification strategy with ever-improving delivery systems, I think, could meaningfully shift what's possible for gene therapy in hard-to-reach tissues.